applications in basic and fundamental research. Sir, over to you.
Thank you very much for the introduction. Uh, I'm Anshuman. I'm from University of Kalyani, and today I'll be talking about bioinformatics. So I'm going to share my screen with all of you. First of all, am I audible to all of you?
Yes, sir, you are audible.
Okay. So I'm going to share my screen with all of you.
Safer.
It's visible. Hello.
Yes, sir, it is visible.
Okay. So let us start. Introduction to bioinformatics. So I'm not sure how many of you are familiar with bioinformatics. So I'm starting from the very beginning. So what is bioinformatics? Bioinformatics is actually it is multidisciplinary science. It involves biology, it involves environmental science, it definitely involves statistics, mathematics, computer science, as well as biochemistry, molecular biology, biophysics, chemistry. So it is kind of an amalgamation of all the different topics, all the different science subjects. So the basic definition. So people are generally confused about bioinformatics and computational biology. So bioinformatics basically is the research and development and application of different computational tools. That means the tools that are available, the tools that are available online, the tools, the pipelines that are available in the different labs and so on. And computational biology is basically the analysis, is basically dependent on the application of the tools to analyze the various types of biological problems. So finally, the bioinformatics and computational biology, they generally like the science of bioinformatics and computational biology, they generally go hand in hand. And so it is basically the solving the biological problems, whatever the problem may be. So by solving the biological problems or problems pertaining to various aspects of biology with the help of computers and computer science. So basically, what bioinformatics, as you all know, it is a very young subject, and that means initially the bioinformatics started in the beginning of 2000. So after that, we are specifically concentrating on spreading awareness of bioinformatics. So one of the important aspects, that means bioinformatics is typically a research based subject. Mainly the main focus of bioinformatics is research. So there are two kinds of researches. One is the fundamental and basic research, and the other one is the research that are being carried out in different industries. So one of the major aspects of bioinformatics research is the molecular bioinformatics. What is molecular bioinformatics? Molecular bioinformatics involves the use of different computational tools to analyze the information present in the different types of biological systems. For example, the system may be a one-dimensional system, like information. It can be the two-dimensional information, or three-dimensional information, or multi-dimensional information. So one-dimensional information means it is basically the sequence level information. Two-dimensional information means how the sequence is arranged in the two-dimensional space. Three-dimensional information means the final arrangement of the two-dimensional data in x, y, and z coordinates. And the four-dimensional means it is basically the real life data. That means if we are working with real life subjects. So finally, this is the definition that we all use. It is the branch of science concerned with the information and information flow in biological systems. especially using computational methods in genetics and genomics. Here, we are kind of narrowing down the definition, genetics and genomics. But genetics and genomics involves the major aspects of all the biological fields. So, what the bioinformaticians do, the biologists, what they do is they collect the data, like the DNA or the protein sequences, RNA sequences, gene expression data, and so on. What computer scientists do is, computer scientists that involve mathematicians, statisticians, software developers, programmers, and so on. What they do is they gather the data, they create the databases, and they use the tools to analyze the data. And finally, the bioinformaticians produce the results. Bioinformaticians give you the results of the analysis. That means as a bioinformatician, one has to be proficient in biology, one has to be proficient in running the computer tools, as well as the various, that means one has to have certain amounts of idea about the basic and fundamental disciplines like the physics, chemistry, mathematics. So this is basically the fundamental aspect. Purines and pyrimidines of DNA. Purines like two ring purines, and pyrimidines one ring DNA bases. And what that means, how the DNA bases are present. The DNA bases are present in the genome, and the chromosome contains various types of DNA bases, and the eukaryotic species. There are different kinds of genomes. That means the nuclear genome, mitochondrial, and uh, plastid and bacteria and other cellular, that means lower eukaryotes, they have genomes, they have the linear genomes, and also they can carry plasmids. So central dogma. What is central dogma? Central dogma, it's DNA conversion of DNA to RNA, RNA to protein. Now, nowadays, the bioinformaticians use the second definition of central dogma. That means DNA to DNA, that is the replication, DNA to RNA, that is the transcription, RNA to protein, that is the translation, and sometimes RNA to DNA, that is the reverse transcription. So nowadays, because of that SARS-CoV infection, which is basically involves this RNA makes DNA, this thing. So the next type of basic data in bioinformatics are the building blocks of proteins, like the various types of amino acids. And the amino acids are classified like this. These are the amino acids. These are the charged amino acids, positively charged amino acids. These are the negatively charged amino acids. These are the polar uncharged amino acids. These are the some special amino acids, cysteine, glycine, and proline. And these are the hydrophobic amino acids. All these 20 different amino acids constitute the one-dimensional data for proteins. Now, another important aspect, another important data that are constantly being used in the fundamental research in bioinformatics, that is the codon table. Codon table, you all know the three triplet codons. They are translated into the proteins. That means the amino acids of the proteins, I should say. So this is the these are the different types of codons that are available, and the corresponding amino acids that they code, they are also given. So this information is very much useful in bioinformatics. I will explain how these information are used in the later section. So first, let me show you the different types of one-dimensional data that are used in bioinformatics. Now, and this is an example how this particular type of one-dimensional data are used in bioinformatics. For example, say you have a DNA sequence. So it's the DNA sequence. Now, in the DNA, from the DNA sequence, what can be done is that the DNA sequence can be translated into the protein. How? Using the codon table. For example, first three letters, they would represent one particular amino acid. The second one represent another amino acid. The third one would represent another amino acid, and so on. So while considering the translation, it is called in silico translation or bioinformatics translation. We need to convert the T's with U's because DNA is not directly translated into proteins. We all know that the RNAs are translated. So that is why, since RNAs do not contain the T's, that is why these T's are converted to U's. So that is the use of this codon table. Like now let me give you a real life example. Say you were working in a lab, and from different experiments, what you identified is you identified the complete genome of an organism. Now you just have the complete genome. You have no other idea about what the complete genome does, what is the reading frame, what is the ORF, and all those. So using that particular codon table, you can identify, at least you can not identify, I should say, you can at least predict what is the reading frame, what is the ORF, and what type of amino acids might be obtained by the translation of that particular region of the genome. So this is routinely done in different bioinformatics labs. Now, this is the secondary data. This is the structure. That means you all know this is the structure of hemoglobin, human, and it contains these helical regions and these sheet regions as well, and loops. So this information, this structure, secondary structure information is the secondary data. Now, how a computer program reads it. Computer program reads the sequence like this, using the single letter code. Now, this is the ORF, this red portion. This is the start codon, this is the stop codon. And using this entire sequence, entire reading frame, this protein is obtained, the amino acid sequence. This is the sequence of the whole genome. And how big a whole genome can be. For lower eukaryotes like E. coli, it is approximately like there are 4,000 genes and around like 5 into 10 to the power 6 nucleotides. Yeast approximately 15 to the power 6 nucleotides, having nearly 6,000 genes. In human, it is in the range of 10 to the power 9 nucleotides. And approximately, we have nearly 30,000 genes. The human genes are present in the human chromosomes, and the smallest one is in the order of 10 to the power 6 nucleotides. So, yes, this is a kind of that means I think I mentioned this before. So first, the bioinformaticians use the sequence information, which are coming from various wet lab based experiments. So from this wet lab experiment, we have the genomic DNA, we prepare the library, and then we clone them and get the sequence. So It is called a UCMSC genome browser of human. It stores the genomic data. So one of the fundamental aspects of bioinformatics is storing, and the data, the different various types of data are stored in the different types of databases. One of the important databases is the UCSC genome browser. Next, using the genomic data, what we can do is we can analyze the data to identify the molecular evolution. This study appeared in Nature. Here, this is the reference, and this is the evolutionary analysis of the H1N1 influenza virus. Now, the gene expression data. This is the gene expression data, and there are 39 genes here available between two classes of tumors. One is Wilms tumor, and the other one is the regular tumor. And so the gene expression data can also be used to study the various types of tumors as well. This is called the heat map. And this red region represents once the tumor is removed, what is the probability that the tumor will come back. So the red region represents the tumor will not come back, and the relapse region represents the tumor might come back. And now the analysis of the regulations. There are various types of DNA binding domains available, and what the DNA binding domains do is that the proteins contain the DNA binding domains. What they do is they might directly bind to the DNA to activate certain biological processes, or as well as they might bind to the DNA to deactivate the biological processes. So it depends on the biology that we are studying. We can channelize the bioinformatics experiments in the different aspects. Now, okay, so far I discussed about the primary data, analysis of the primary data. Now, I'll be moving towards analysis of the secondary and tertiary data. So before I move, we can have a couple of questions. I can have a couple of questions. If you want me to clarify anything, you can ask me. The audience, if you want to ask me anything pertaining to the data that I showed you, so you can ask me the questions right now. So shall I move on?
Yes, sir. Maybe no questions.
Okay.
Any person can unmute and ask the questions.
Yes. Sorry, I can't hear your voice. I can't hear your question. So did you ask any question?
Any person, or at least you can raise, or you can put the question in the chat box so that I can read it.
Your voice is breaking. I can't hear you. So can you please write it down in the chat line? how the protein helps in diet. This is your question. Okay. So how the protein helps in diet is a very big question. For that, you have to have some idea about the biochemical pathway where the protein is involved. Now, in the biochemical pathway, say the protein that you are interested, your protein of interest, is found to be interacting with certain other proteins. And that interaction leads to the onset of, say, a specific metabolic process. Now, you can study that protein binding interactions to analyze how the protein or which amino acids of the protein are interacting with its partner, so that in the cell, the specific biochemical metabolic reactions can be carried out. Now, if there is any problem, or if there is any mutation, or if there is any change in the protein structure, then that altered protein might not be able to interact directly with its binding partner. So that way, that might lead to some blockage of the metabolic reactions to which the protein is associated. So that way, you can answer, you can find out how that protein of your choice is involved, like is utilized in metabolic reactions, those metabolic reactions that are very much essential for our body, and the protein of your interest should be consumed from external sources. That much can be done through bioinformatics. Anything else? Any other question? If there is any question, you can write it down in the chat line. If there is any other question, you can write it down. Okay. Your question is how bioinformatics facilitate to characterize drug resistance and its side effects. Yes. Bioinformatics can be used for that. That is, here also we have to have the target. For example, say P53. It is a specific protein which is associated with many of the biochemical reactions. Now, if you want to block the P53, in that case, you need to have the structures of certain molecules which have the abilities to bind to P53. Now, under certain circumstances, there might be some mutations in P53, so that the molecules that you develop, that you generate, those molecules are no longer able to bind to P53. So that way, we can consider this to be a drug resistance, a mechanism of drug resistance. And side effects can be obtained by calculating the ADME properties. Absorption, dissolution, its mode of action, and excretion, ADME properties of a molecule. if there is any change, that means if the molecule of your interest, that means the molecule that you are considering to use as drug, if it cannot be administered, that means if you cannot take that in your cell, that is kind of, it's one of the side effects. Once it enters the body, the molecule might interact with certain other molecule. That means there might be multiple targets for the molecule that you are using, that you are trying to use as a drug. So that multiple target interaction is another aspect of the side effect. So that way we can calculate. But whatever the case may be, you have to have the structures or the information about the target and the receptor and the molecule. Anything else? Anything else?
I think, sir, you can continue now. There are no further questions, I see.
Okay. Okay. Then the next portion, that is another fundamental aspect of bioinformatics, is the structural bioinformatics, that is the protein-protein docking. Protein-protein docking means it is the identification of the binding interactions between a protein and a target. Now, the basic idea is, first, you are having the genome sequence. It is translated into protein, an amino acid sequence. From the amino acid sequence, you can build the structure of the protein. That is called homology modeling or model building. And then the model protein can be used to find its binding interactions with various types of other binding partners, like the drugs, small molecules, or another protein. This is the time frame of identification of a genomic sequence. Then we can perform sequence alignment. We can use this protein data bank, which stores the structure. We can use the gen bank, which stores the sequence. And these are the different other tools. NCBI is one of the most important and vital database or storehouse or repository of genomic data. FASTA is a specific format which is used to store the data. That means the FASTA format is most universal and most widely accepted data. BLAST is a tool to analyze, to compare the sequences. And SwissProt is another database which stores the information about proteins only. NCBI stores information about both proteins and nucleic acids, and SwissProt is specific for proteins. Okay. Now, I'm skipping this. Okay. So, GenBank, NCBI. GenBank stores the nucleotide sequence. Initially, it used to store only the nucleotides, but nowadays it stores the information that are derived from nucleic acids, that means the amino acid sequences of proteins as well. So this is an older statistics. So now the next one, that is another basic aspect of bioinformatics, is annotation. Annotation means what information can be derived from the primary sequence data. The information can be the open reading frame, can be the functional site, can be the structure and function. That means, say, let us take an example. You have the genomic sequence. From the genomic sequence, you can use the codon table to identify the open reading frames. From the open reading frames, you can use the bioinformatics in silico translational techniques to identify the amino acids that are being coded by the open reading frames. The amino acids that are being coded by the open reading frame can be analyzed to identify its structure and possible function, or as well as the location of the active sites. That is called the functional sites. This is an example. Another. This is the promoter region. This is the transcription factor binding site. This is called the ribosome binding site, and these are the open reading frame and coding sequences. So all these are called the annotations, which are obtained by analyzing or comparing the sequence with the existing other known sequence. Say this is an unknown sequence, and these annotations can be obtained by analyzing or comparing this unknown sequence with the known sequences that are present in the database. Comparative genomics. As I told you earlier, that the entire process, the entire mechanism is obtained by comparing the sequences, comparing the nucleotide sequences, comparing the amino acid sequences. The major aspect of bioinformatics is to have the annotations. That is because a mere sequence, that means a nucleic acid sequence or the amino acid sequence of a protein, just a mere sequence, does not give you any idea about anything. Until and unless you have the annotations, you perform the annotations, those sequences are meaningless. So you have to use these comparative genomics techniques to identify the various types of information stored in the sequence, inside the sequences, and that is called the comparative genomics. Uh, okay. Now, an important aspect of comparative genomics is the alignment. Say, in this example, we are aligning the pre-pro-insulin from Xenopass and BOS, and we are aligning the sequences like this. This star represents the region where there are perfect alignment. These double dots and dots are represent the synonymous substitutions. And these are the gap regions. Gap region means these are the mutations. See, these boxes would represent the most conserved regions. This is okay. There are a certain I'm showing you this paper because for every genomic scientist, that means the bioinformaticians who are working in the field of genomics, this is a very textbook kind of paper. It represents the various types of regions that are present in the human genome. So here, one important aspect is that majority of the portion of the human genome is found to be non-coding. Those regions were considered to be junk regions. But nowadays, with the advancement of techniques, sequence alignment techniques, as well as sequence identification techniques and next generation sequencing um, techniques, we observe that those junk regions would provide many more information than the coding regions. Those regions are mainly referred to as non-coding regions. Sometimes the non-coding regions are classified as long non-coding RNAs, short non-coding RNAs, piRNAs, miRNAs, etc., and so on. So next, we move on to the functional genomics. Functional genomics means this is the genome wide profiling. We have the mRNAs, we have the proteins. Now, what we try to identify is whether the proteins that are obtained from mRNAs and those proteins are co-expressed. Also, we need to identify the protein-protein interaction for network analysis. So this is the function of the genes, and the other part, this is also called the heat map. This is Saccharomyces cerevisiae. This is microarray chip. These bright regions would represent the co-expression or protein-protein interaction. Now, the next fundamental aspect of bioinformatics is structural genomics, to identify the structures. So, for structural genomics, we need to have the help of SwissProt and PDB. Now, the various types of databases. Databases means this is another aspect of basic bioinformatics. It is a database is a large structured collection of data, and all these data are stored in databases in a user-friendly manner. So, okay, I need to show you this one. This is called the flat file format, but mainly we use the accession number. This is now and the different types of data that were used to be stored were in CD-ROMs, in computer tapes, floppy disks, but nowadays most of the data are being stored in WWW, that means World Wide Web. And this is a kind of statistics. More than there are 1,000 different types of biological databases which store DNA, protein, RNA, their 3D structures, and all this information. These are the different categories of life science databases: sequence databases for DNA and RNA, genomics, mutation polymorphism data, protein domain and family information, structure information, and literature databases, and so on. And these are the URLs of the databases that are frequently used in bioinformatics. Now, the literature database. Literature database, bookshelf. Bookshelf is a collection of free online books. PubMed. We all know, we all use PubMed in a regular basis. OMIME. OMIME is online Mendelian inheritance in man. It's a database of genes and genetic disorders. This is a screenshot of NCBI, and this is line PubMed literature databases. This is of PubMed. which gives you an idea about how to search it. You can use any of the keywords here to search PubMed. This is the abstract of PubMed. And this is the way, this is how you search the different types of query, different types of data in NCBI. You can use any of the syntax. You can use Boolean operators. You can use literature. You can use text, anything. Sometimes the query, you need to refine the query because say you are using the keyword hemoglobin. That would give you the data pertaining to all the types of hemoglobins in all the different organisms. But once you restrict your data to human hemoglobin, you get information about only the human hemoglobins. And these are the other important terms associated with it. So the most important one is BLAST. Skipping this. This is a screenshot of PubMed where you can limit. You can use all fields. That means that would use the keyword to search both the author names, the date, the first author, grant number, anything, and the subsets. In a similar way, you can search it. And these are certain, you will, once, if you would like to work in the field of bioinformatics, you should be familiar with all these synonyms, all these abbreviations. Like all fields are sometimes written as all, journal title is written as TA, pagination, PG, and so on. So you should be familiar with that, with all these terms. This is a generic one, and this is a bookshelf. So you need like this is I need not have to explain this one because all of you are constantly using the various types of databases, so you know how to search the databases. So searching up PubMed means you can use like this. You can use the terms like this. You can use these. That means you can use quotes, stars, something like this. You can store the history in PubMed as well, search history. So now next comes OMIME, Online Mendelian Inheritance in Man. Let me show you. OMIME has certain categories. For example, the result with a positive sign gives you the gene with known sequence. Hash means a phenotype description, and so on. So you can use these keywords to search OMIME. Again, these are the abbreviations. This is OMIME. There are other literature databases like Google Books, Google Scholar, Web of Science. You can use all those databases to carry out your bioinformatics task. So these are very specialized ones, so I'm not going through the details of this. And this is a kind of a website. Generally, the beginners, like the students or the researchers who would like to start bioinformatics research, these websites would be very helpful. So you can look into these websites that would give you these websites would give you certain ideas about the various aspects of bioinformatics. So this is Google Scholar. I'm skipping all this because these are very specialized ones. You need not have to like use all these because there is PubMed. You can use the Google book search as well. It is very simple. So this is the different aspects of bioinformatics, the fundamental and basic aspects of bioinformatics. So to summarize, what I would like to say is that the bioinformatics uh, basically is dependent on the data that are stored in the different types of databases. The data can be the sequence, can be the structure, can be the literature, anything. According to your problem, you can choose the data as well as you can choose the tool so that uh, you can carry out your work smoothly. But which data to choose or which tool to use is dependent on the problem, is dependent on the problem that you are facing, that you are trying to solve. So, and another important aspect is that bioinformatics is very much dependent on research. So, this, as I told you earlier, that the subject is still very young. So, you won't find too many. And now you can find a couple of good literature, good books, but previously there are no books or data or journals available. So it is always good to be familiar with the upcoming topics and the upcoming fields in the bioinformatics. So now I can take your questions. I can take your questions now.
Anybody, please ask questions if you have any doubts.
Or you can type in the
You
chat
can
line.
type the question.
The voice is not very clear.
You can type in the chat box the questions for sir.
No, you cannot. Eyeball is just to view the protein structure. For validation, you need to energy minimize the protein using phosphorus.
Any other questions, please? If you want, you can unmute also and speak. Sir, there is one more question in the chat box.
Yes, I see. I'd like to know the data that you have selected. What is confound in that? Can you please be a little bit more specific what you want to know? There can be various types of data. The sequence, the structure, and the literature.
Divya, if you want, you can unmute and speak. Divya, if you want, you can unmute yourself.
Yeah, hello, ma'am. Hello.
Yes, Divya, you are audible. You can ask your question.
Yeah, okay. Ma'am, I would like to know what is the confound? Like in the sequence analysis, you gave the examples of tumor in the previous slides. Hello.
Yes.
Yes, in that you have given the example of tumor, like green color, which shows that tumor can be treated, or red color can be treated, and green color which cannot be treated. Like on what basis? Like it is age wise, or something like whether that whether it is related with the disease, if the person is infectious, uh, that a tumor, it is the infection is associated with an protein which giving rise to a kind of cancer. So in that case, what is the confounder in the data?
The data that that are generally used are the patient sample data. They are mainly the protein interaction data. So some of the protein interactions, they are irreversibly damaged. And if the protein interaction are irreversibly damaged, there can be a relapse of that particular disease. But if it is damaged reversibly, in that case, if we remove the problematic part, we can cure that particular disease. So that is what is shown here. Whether the damage is reversible, that means whether the protein interaction is irreversible or reversible. That means whether the two of the binding partners, whether they are interacting in a stronger way or in a much weaker way.
Okay, ma'am.
Any other questions? You can unmute and you can ask.
Phytochemical analysis. Yeah. Recently, lots of phytochemicals are constantly being analyzed. So phytochemicals means these are certain small molecules. And first, bioinformatic techniques are used to analyze the functional groups and the characteristics, the properties of the functional groups that are present in the different phytochemicals. Now, with the help of bioinformatics, the ADME properties are checked. ADME properties, I told you that absorption, dissolution, mechanism of action, and excretion. What are the different parameters? What are the different values of all these? whether that particular phytochemicals can be easily absorbed, whether that particular phytochemical can function, can work inside the human body, whether it can be after its function, whether it can be excreted from our body. All these properties are to be checked by bioinformatics. Then, using molecular docking techniques, the phytochemicals can be used to identify the binding partners. That means to which protein that particular phytochemical has the most affinity for.
क्लास में बैठ गई तो क्या करूं रुक जा भाई your audios. If you want to ask questions, you can.
You will find most of the Chinese companies and Chinese groups, they are using phytochemicals to treat the SARS-CoV.
Any further questions?
Any other questions, please ask, or you can type it in chat box. Sir,
Yes.
hope there is no further questions.
Okay.
Uh,
Then thank you
if
very much for listening to this presentation. And my email ID is there. So if you need any help, you can email me your queries. I'll try to help you.
Thank you, sir. Thank you so much for your wonderful talk on bioinformatics. You have really explained very well about bioinformatics and its applications. Thank you so much, sir.
Thank you. So,
Uh,
uh, and before I conclude, I would like to say one more thing. In our university, University of Kalyani here, we have a bioinformatics infrastructural facility center, and this is there to solve the bioinformatics problem. So if you have any bioinformatics related problem, you can send me your queries. We'll try to solve or try to give you answers of your queries using that facility center. It is the DVT funded center. So yeah, I think there is how to perform gene homology. Okay, chloroplast, that is simple. You can take the sequence, any protein from chloroplast, and perform a blast search. From the blast search, you will be able to identify its homologs. Um, okay, so I think there is another talk, and the next speaker might be waiting. So if you have no more doubts, so I can conclude here.
Yes, sir. I think we can start the next session.
Okay, so
Okay.
thank you very much.
Yeah. Thank you. Thank you so much, sir.
Thank you, sir.
There is one more question, I think. Anshuman, sir.
Oh, one more question.
Yes. Uh, just
Description, transcription, translation. No, transcription, translation, because the data that are there, those data are obtained after transcription and translation processes. So these techniques or these processes, these biochemical processes do not matter. Okay, so I think we are done for the day. And any other question, any more questions or any more doubts, you can send me an email. So all the best to all of you. Thank you very much.
Thank you so much, sir. Thank you. Uh, Next, for the next talk, I would like to invite Dr. Prinon Bakchi, Assistant Professor and Researcher at Padma Shri Institute of Management and Sciences, Bengaluru. He has completed his postdoctoral research from the University of Lorraine, Nancy, France, and from Science and Engineering Research Board, Government of India. He has done his doctorate from Career Point University, Kota, Rajasthan. He holds certification in Ayurveda, Yoga, and Sanskrit. During his doctoral research, he had attended IPRO sponsored Advanced School of Neuroscience at Monash University, Malaysia. He has authored over 50 research and review papers in several national and international journals and in conference proceedings and presented his research works in conferences abroad. He has guided several graduate students. He is an editorial board member of International Journal of Public Mental Health and Neurosciences, the International Journal of Psychosocial and Cultural Genomics, Consciousness and Health Research, and Neuroscience Research Letters. Currently, he is an intern under Department of Health, Government of India. With this brief introduction, I will welcome Dr. Prinon Bakchi for his talk on latest tools and trends in literature survey. Sir, over to you.
yes. Thank you so much, madam, for your wonderful introduction. So I will not. I will directly start with the tools and techniques because Anshuman sir already has given the brief about bioinformatics and he has showed the tools. I will be demonstrating the tools now. So give me some time. I will share the screen. I am trying to open the uh, tools for your reference. Give me a moment. Is my screen visible to all?
Yes, sir. It is visible.
Okay. Okay, great. Yeah. So I think sir was talking about this NCPI database. Yeah, I won't say it is a database, it is a rather a master database. Whatever information you need for biological sciences, everything is available here. Everything. You can see here in the drop down menu, you can see here all databases, assembly, bio collections, bio project, books, whatever you want. It is given even you have PubMed. I think sir was explaining about PubMed. SRA, this is for the next generation sequencing analysis. You have the data. You have the data here. Taxonomy, and there are several tools available. So it is in other one sense it is a master database. It is a collection of several databases. So suppose if you want to retrieve any particular information, say for example protein. I have selected one protein for you guys. So from here, this drop down, I have selected the protein and randomly I have taken one protein, that is, say for example, hemoglobin. So you can see here there are 1 to around 20,000. It is not 20,000. It is 2,09,458 data are available here. In your right hand side, I think you can see my cursor moving, right? So you can see here there are several other taxonomy. So whichever organism you want, you can see here. If you are a particular organism, you can select from here. Say for example, you are specific only to this Homo sapiens, like for example. So you can click on that. You will be getting only the Homo sapiens sequences. Where did I click it? Okay, yeah. So you can see here the all the out of the 9 lakh sequences you have 2,737 Homo sapiens sequence of hemoglobin. So for your reference, I will just click on the first one. Anyone you can open. The information will be. I will show you what are the informations available here. So first lines are the gene identifiers, and you can see here it is like having 105 amino acid sequence. It is a linear, and it was published online on 14th of July 2016. And this is the database source from which particular database is this particular sequence retrieved. Then you have the source organism. Then it is like source examiner, that is the human sequence, and this is the classification, how it is coming. And there are several references in that this particular authors they have submitted it, but the data is still not published. If it is published, the journal title and the other details will be appearing here. So this is reference number 2, and this is direct submission, and you can see that this is published in medicine journal, and this is the particular I think this is not journal, it is just from the institute it has, they have submitted it. Okay, so this is your protein sequence, this is the raw data. So if you want the data to be analyzed, you have to click on this FASTA. If at all your particular sequence is having any 3D structure, it will be displayed in your right hand side. You can see here protein 3D structure. So this particular proteins 3D structure is already available in the database. Which is the database? That is protein data bank. I will come to that. So this is your sequence. It is the FASTA format. You can either copy and paste it in Notepad because you will be using this. There is other option. There is one option like send to files, download item, and create file. So there is two options like one is that you can even open in a notepad and you can do it. Other option, you can either choose this option, send to file and download format FASTA and create file. Either ways you can do it. So then next option is sir was even telling about BLAST. So what is BLAST? You can run the particular sequence using BLAST here, or I open the window for you. I will show you this. Yeah. So I think you all can see this particular screen of mine. So what is BLAST? BLAST finds regions of similarity between biological sequences. The program compares nucleotide and protein sequence to sequence database and calculates the statistical information, statistical significance.
Thank you, Anna.
So there are actually there are 5 types of BLAST, but here I can see only 3 types. Other 2 are not there. One is this one BLAST N. This is nucleotide BLAST, that is BLAST N. Other is protein BLAST. Other is T BLAST N. One more is T BLAST X, that is not here. BLAST X and T BLAST X. Those 2 are not here. So we can see here what is this first. Anyway, we have taken the protein sequence. We will go with the protein BLAST. In protein BLAST, what is the criteria is your input, the submission will be your protein and outcome will also be a protein. In your nucleotide, it is the submission will be a nucleotide, outcome will be a nucleotide. In T BLAST 10, your submission will be a nucleotide. And it will be, the software will be translating it into a protein query and your output will be a protein. So let us start with this protein BLAST. Just bear with me because I am using Zoom also and internet also, so it will be it will be little slow. Okay. So here you have to put the accession number, GI, that is gene identification number, or FASTA sequence. So already we have the FASTA from the for our hemoglobin. Then copy it as it is. Now, what is this query subrange? If you have any doubt, you can stop me anytime and you can speak. If you have any doubt, you can always unmute and speak. If you are typing in the chat box, I may not be able to see now. So after each chat box, you can do it later. If you have any doubts, you can stop me immediately. So queries subrange means you want this is how much this 100 what was the 103 or 109 amino acids were there. Yeah, 105 amino acids are there. So if you directly do the blast, complete 105 amino acids will be displayed. But if you don't want, I want only between 10 to 100. So only the software will be searching only between 1 to 10 to 100 amino acids only. Then you can there is the option for uploading the files also. Yeah, otherwise either you can upload it or you can paste it. Upload or paste. I have already pasted it. Then you can put any job title. Already it is taken. If you want, you can put a separate title. Then there is option for aligning 2 or more sequences. If you want, you can use this option. So you have to upload or give one more input. Then you have your database here. You can see here there are non-redundant means all the databases available. Then these are all the specific other databases. So I will go for the NR database. So if you want any particular organism to be searched, example Homo sapiens, you can enter it. And there is one option that exclude. So if you click on this exclude, all the organisms will be searched apart from Homo sapiens. So delete it. Just for example, I showed you. Then there are options like blast, quick blast P, and several others. These are all like repeated blasts. So let us see, choose this option blast P. And if you want in a new window, it will be displayed, or you can even click here. So any doubt if you have, you can let me know. So it will take some time. So we have record the output. You can see here there are so you have so many like distance tree, multiple alignment, and so many others. We will see it one by one. So you can see here these are the several related organisms which has come, and you can see here what are the identity percentage. So you can see here the first few are all 100%. Then again you have 99% and so on. Then there is something called graph summary. Somebody is annotating the screen. Stop annotation. Anybody who is doing it? Stop the annotations in the screen. Yeah. So you can see here the various color codes. So you can see here different color codes will be there. We will please stop the screen.
Sir,
Please stop.
sir,
Who is doing?
disable, sir.
Sorry.
Disable the annotation, sir.
Yeah, you have disabled it.
So, he is doing bioinformatics. He is doing bioinformatics. He is doing some search, translational, transcriptional search. What is that required? Don't know.
Silence, everybody.
Not the LR.
നമ്മളാ ഫ്ലിപ്കാർട്ടിൽ നിന്ന് മേടിക്കരുത്, സ്വന്തം കടയിൽ നിന്ന് മേടിക്കണം എന്ന് പറഞ്ഞാൽ വാട്സപ്പ് അല്ല, സ്വന്തം അല്ല, നമ്മുടെ ചുറ്റുവട്ടമുള്ള അവരൊക്കെ വലിയ കോടീശ്വരന്മാരല്ലേ, നമ്മൾ
Mute your audios.
Silence, everybody.
ആൾക്കാരല്ലാതെ
Silence.
നമ്മുടെ കടയിൽ നിന്ന് ആരും മേടിക്കരുത്, ഫ്ലിപ്കാർട്ടിൽ നിന്നും. അതെയാ,
Please mute your audio.
So you can see here. You can see here various color code options are coming. I have disabled the options, so nobody can unmute yourself now. It was difficult. I don't know what happened suddenly. So there are various color codes. So once the scores are decreasing, the different color codes will be there. Or you can say, I won't say even the scores here, maybe the identities. So these are all around 100, 90% identities will be here. Little less, around 80% identities will be having pink color. You will be seeing if it is subsetting class size, subsetting I will show, then it will be there. Then you will have light green color. And further like that, you will be getting this graphic analysis. The next part is alignment. Alignment. Somebody was asking like how the alignments and how the regions will be shown. So you can see here, the first you can see here, like identities is 100%. So there are no gaps. Both the sequences have aligned without any gaps. So there is no space for any extra or additional sequences. So I will show you the differences. In the below sequence, it will be there. So suppose you can see here identity is 99%. So there is a small mutations occurring here. So like this, you can see the differences in blast search. Here again, there are it is 99%. You can see here, this is one mutation is occurring here, and so on. In this manner, you will be seeing the alignments in blast search. There is one more option, like tree. So you can see that in using BLAST also you can generate the trees. Such kind of tree you will be seeing in so many research papers. So example, so this particular organism and yeah, here this particular organism and this particular organism is quite similar. Again here this particular organism and this particular organism is similar. In this manner you can see it. So this is your tree view for the various sequences which the BLAST has searched. Okay, so this is one part. Next part I will show. I will tell you about protein data bank. If you have any doubts, you can put it in the chat box. So this is your RCSB. It is also maintained, or it is a part of your NCBI. The RCSB PDB is also a part of your NCBI database. These are the statistics of RCSB PDB. So it has around 17 lakh 5,097 structures, and it has around 49,744 structures of human sequence and 12,456 structures containing the nucleic acids. So complete statistics are here in this protein data bank. Suppose how you can see the structures in your initially when I was showing you this hemoglobin, I had told you in your right hand side you have your protein structure, 3D structure. You have your PDB ID. I will just highlight it. So I have highlighted it, so you can see here PDB ID 4HHB. Same ID you can type it here. So you can view the information about the protein here. In your left hand side, you can see the 3D structure and several other information about this particular protein. So if you can visualize either from here or there is an option like download PDB format. So this is your 3D structure. We can see, and I have used the software Rasmol for visualization. Then there are several other. You can see it like you have wireframe, you have backbone, sticks, space field, and so many others. Usually in the research papers, you will be seeing this ball and stick, which is very common. Even the others are common, but this is most widely used ball and stick model. Okay. Now there is one more a software or database that is called as PubChem. In PubChem database, there are all the phytocompounds, which this is the database for all the phytocompounds. Example, like any phytocompounds you can recall, like for example, curcumin. Curcumin, it is a compound found in turmeric. So somebody was asking about the phytocompound. So this is the database. This is one usually widely used. The database is PubChem. So this is, you can see the best match. There are so many other structures available. So we'll see the first one. So this is the various information available for PubChem. Sorry, yeah, the curcumin 2D structure is here. Even 3D structure is here. You can see here there is a different format, but we generally need the 3D structure. In this will come in the SDF format. I will show you. So this is your 3D structure. So usually what we will do is we will download this. There is an option like download. Once when you are clicking it, there is an option like SDF. So this one you have to save it. This SDF format will not open in your Rasmol. So how to open this SDF format in the Rasmol? So there are several softwares available, and even sometimes I can do it even online. There is something called as molecular file format converter. So this Open Babel is one of the software, online software, wherein you can convert the file format. As I told you, the PubChem gives your results in SDF format. PubChem gives you a result in SDF format. You can convert it into PDF format using, it's not PDF, it is PDB format. For PDB format using this. An option to become, I don't know why it is not coming. One minute. There's some problem with the software. Yes. Yeah, this was not coming. I don't know why. Now it has come. So your input, as I told you, we had downloaded the SDF format. So this is your SDF format, and the output, as I told you, need in the PDB format. So this is your this one curcumin SDF format. Convert. So this is what you are getting in the PDB format. So you can save directly here. Save as. Don't know if you can see the notepad. So here I have pasted the protein data bank coordinates in this notepad. Save as. Suppose I am saving it in the desktop. Change it to all files. Curcumin.pdb. So it is saved. So now you can see this curcumin in Rathmon. So you can see your curcumin. We have we took the SDF format from the PubChem and we had converted into PDB and we can visualize in the Rasmol. So as usually I have shown you last time, there are various formats available. So as I told, ball and stick is the widely used. So you can use that. Then I will be opening this for docking. I have little time. I will do the docking for you. Will just show you. So one of the docking software is PatchDock Server. I think sir was even discussing about this docking. So I will show you how the docking works here. So this is one of the software. This PatchDock is one of the software for docking. So you have to input as I sir had told you, you have to there in docking there are two molecules. One is your protein and other is your ligand. So receptor molecule is your protein molecule. So this hemoglobin you can see here, this 4-HHB, this one can be your receptor. So take it receptor molecule, and ligand molecule is your curcumin, which we just now we took from the PubChem. So let us select it. I had saved it in the desktop. So you have to type your email ID. I am typing my email ID and keep everything as default and submit. So it will come in my email ID. I will show you the analysis. It will take some time. It will come. Yes, it has come. So you can see here the receptor 4-HHV and the ligand curcumin has docked. So let us open it. So you can see that this is the docking complex. I will change the format to this wireframe. No, there are two chains rather. You see that the red molecule is your curcumin here. Because the command which I gave, it will be like other molecule will be blue and the compound will be your red. So this is how the docking will be, docking analysis will look like. To see in a better manner. Since you have taken a molecule from the protein data bank, it will have so many ligands also. So you can ignore the other ligands. Usually in your docked complex, the ligand, whatever you are taking, it will be in the red color in your rasmol. Okay. Okay. If you have any questions, you can ask now. I will unmute. I will unmute all. Yes, you can ask the questions now. I have unmuted you all. You can unmute and you can speak, or you can put it in the chat box if you have any questions. Any questions anybody is having? You can unmute or you can put it in the chat box. Anybody have? You can ask the questions now. Am I audible to all?
Yes, sir, you are audible.
Okay. Okay, if anybody has no questions, then we can close the workshop. Is the main importance of RCLP peer review research? Yeah, main importance, it is the database. It contains the 3D structure of all the proteins. How to read the scores? Okay, very good. How to read the scores? Yeah, this is the score. It is called the docking score, or it is also called as your NRE value. This is usually in the negative. So you have to read it like minus 5062 kcal per mole. That is kilocalories per mole. It is in negative. Negative means it is the negative potential energy of the complex. It is not the stability of the complex. So that is how you can see that you can see it is an increasing order. It is not actually increasing. It is in a decreasing manner. So it is in the negative. So minus 5062 minus 4996 and so on. Yes. Did everybody understood the score, Anumbia? Did you understand? Or you want me to repeat it? I think there is some problem. Okay, so yeah, I think my voice is clear now. Yeah, so I was telling about the scores. I told you these scores are negative. So it is like minus, you have to read it like minus 5062. It is expressed in kilocalories per mole, kcal per mole. So it is like negative energy or negative potential energy. As you know that once the potential energy should be minimum, so then only a complex can be stable. So the energy value should be as low as possible. So this is minus 5062, and then it is in the increasing order. So this is what your score signifies. Any other questions? Any other questions? Please let me know.
Sir, I think no more questions, I guess. If anybody is having any questions, you can
Yes.
ask. Anyone wants to ask any doubts or anything to discuss, you can. Or you can type it in chat box also.
If there is no other further questions, we can conclude the session here. Madam, we can end the session now. I think there are no further questions I see.
Yes, sir, I guess no questions.
Yeah. Yeah.
Okay, sir, thank you so much for this wonderful session. Thank you, everyone. Thank you so much. We will continue with the tomorrow's workshop with some other topics. Thank you, everyone. Sir, I think you can end it, sir.